Powdery mildew of the vine is caused by the fungus Heliosulphore necator, which has the characteristic of extending its mycelia along the surface of the leaves. Spore-carrying conidiophores then emerge. The combination of mycelia plus spores causes the whitish-gray powdery symptoms characteristic of this disease. The asexual spores of the fungus, also known as conidia, are easily spread by the wind. As soon as spring arrives, the conidia infect the leaves, causing new sporulating patches. At first barely discernible, the disease develops rapidly on the leaves, producing large quantities of conidia which will contaminate the future berries. Now let's take a microscopic look at the growth of a conidium which has landed on a vine leaf and infected it. As soon as germination begins, the conidium forms a special organ called the appressorium, whose function is to anchor it to the leaf. Once it is securely anchored, it extends its first germinating filament, then forms secondary filaments. 5 days later, the conidium has produced a vast network of mycelia on the leaf from which conidiophore stalks will emerge progressively, carrying with them new conidia. Then a dense forest of conidiophores appears. Only 6 to 9 days after the arrival of the conidium on the leaf, the next generation is ready to be released and migrate to another leaf and produce a new infection, perpetuating and multiplying the disease exponentially within the field. Let's stay in the microscopic world a bit longer and observe how Heliosulphore S acts on the biological activity of the parasite during each phase of its cycle. It holds a few surprises for us, as we shall see. To evaluate the preventive effect, vine leaf discs are treated before infection, either with Heliosulphore S or with a micronized reference sulfur at a suboptimal dose good for laboratory observation. 24 hours after treatment, the leaf discs are inoculated with Heliosulphore necator by shaking a leaf contaminated with conidia over them. They are then put into incubation and observed 9 days later by using 2 fluorescent dyes, a green dye revealing the parasite's metabolic activity and a red dye revealing dead cells. Comparative observation of the treated and untreated leaf discs speaks for itself. Only the dead conidia, those that haven't or have barely begun to germinate, are visible on the leaves treated by both products. This is in contrast to the multitude of conidiophores that have developed on the control leaves. Now, in order to test the curative effect of Heliosulphore S, vine leaf discs are first inoculated with Heliosulphore necator, then treated with Heliosulphore S, or with the reference sulfur 48 hours later. In other words, right when the germinating filaments are developing. 7 days later, the leaf discs treated curatively show the same images as those treated preventatively. Fungal growth is completely halted, whereas the untreated control is teeming with conidiophores and conidia. Their bright green fluorescence demonstrates the intense metabolic activity of the control. As fascinating as this image of Heliosulphore necator sporulation is, we mustn't forget that it's this endless multiplication that causes the extent of a powdery mildew epidemic. But Heliosulphore S has also shown that it has precious anti-sporulating properties, revealed using functional fluorescence microscopy. For this, leaf discs are inoculated, then treated with Heliosulphore S or with the reference sulfur when the conidiophores are forming, in other words, 6 days after inoculation. 3 days after incubation, the untreated control shows us the now familiar image of the fungus's active sporulation. The reference sulfur shows a slight efficacy by reducing the density of sporulation. However, Heliosulphore S has been shown to be much more active by totally blocking the formation of conidiophores. This is most certainly because of its unique pine terpene and high-quality micronized sulfur-based formulation that it has such remarkable anti-sporulation properties. By combining preventive, curative, and anti-sporulating effects, Heliosulphore S acts effectively throughout Heliosulphore necator's cycle, all the way until its final sporulation phase, which is a determinant factor in the progression of the epidemic. In practical conditions, Heliosulphore S shows its best results when used in a preventive treatment strategy, allowing it to reach the parasite in all of its developmental phases in the field.